It's the part A question. Yes. Did you solve? Okay. Yeah. What about the online students? Okay, option 2. Yes. Okay, fine. Yes, good. So, all of you were able to solve it, right? Online students? Okay, fine. So, then I am not going to explain this, okay? If you have already solved it, so I am not going to explain this question. Okay? Yes. Yes. 383. Everyone got it? Yeah, Sarita? No, Meghna, it's not third one. Okay, you did not solve it. Okay. Yes, it is like, you have to see what it is. Actually, if you see the pattern, you will find the middle digit is actually the multiplication of these two. Okay. So 3 into 1 is 3. 4 into 2 is 8. 5 into 1 is 5. 2 into 3 is 6. Okay. 3 into 2 is 6. 2 into 4 is 8. This one is the exception. 3 into 3 should be 9, but here it is 8. Okay, so that is why it is the odd one out of the whatever series is given. Okay. Yes. Okay. So clear for everyone, right? Online students? Okay, fine. Yes, part B question. Yes. So, exactly, okay, somebody is saying A. No, Meghna, it should be just the opposite of it, okay? See, the condition is both is present. Glucose is present, lactose is also present. Now we have two types of regulation, positive and negative, right? So negative regulation is because of the repressor, and what the repressor does, just a sec. Suppose here is the operon, you have the somewhere here promoter, somewhere here you have the operator. Okay. Now the repressor protein synthesized from some gene, okay, it will go and bind always, okay, when the lactose is not there, okay. But when the lactose is present, it forms allolactose, and the allolactose goes and binds to this repressor. As a result, repressor cannot bind. So when lactose is present, repressor is not able to bind to the operator, okay. Now, what happens to the positive regulation? That is done through the cap, right? Now, what is important for the cap to come and bind over here near to the promoter region? The cap binding protein is the availability of cyclic AMP. Okay. Now, cyclic AMP concentration is usually low when enough of energy is available in the body. Okay. Now, when the glucose level falls, then only cyclic AMP concentration increases. Okay, it is a signal that no more energy is available in the body or in the cell. Okay. Now, here the condition says glucose is also present. That means it should never allow the increase in the cyclic AMP concentration, right? It should be always low when the glucose level is high. So, without much of cyclic AMP, cap cannot alone go and bind. Okay, so this is also not allowed. So both of them won't be binding to the DNA. Okay, so that will be the condition in this case. Now clear? Okay. Fine. So yes, clear for everyone, right? Yes. What about you, Madhumanti? Is it clear to you? Yes. A and D. Okay, yes. Anyone else? Online students? Option D. You mean 4, right? Anjan? Okay. Bhavna, yes. Yeah, what about others? Others did not answer it. Meghna, Harshita, some people, yeah. Option 2. Okay, no, fine. Anyone else? See, here you have to remember that the regulation of the transferrin receptor through the stability of the mRNA, right? So suppose this is the mRNA. The 3' end will have some stem and loop-like structures. Okay, these structures will be responsible for the regulation of the stability. Okay, so this is the 3' UTR, not the 5' UTR. So that means this statement is wrong. Yes. Next, what is the first statement? Iron responsive elements in the iron binding sequence is an iron binding sequence in the mRNA that encodes transferrin receptor protein. So this is what is. So this is the mRNA of the transferrin receptor. Okay. So it will have this particular iron response element at the 3' UTR. So this is true. Okay. Second one is not true because it says a 5' UTR. So this is not correct. Okay. Then it talks about the protein which comes and binds to this particular region. So that is a aconitase, right? So here we can also call it by general name, that is iron response element binding protein. Okay. Now what it is saying is, when iron is abundant, then only this protein is binding and then it is stabilizing the mRNA. That is not true. Okay. Yes, this is not correct. Suppose you have the iron response element. Okay. If it is binding to iron, then it is forming the holoprotein. Okay, and this holoprotein does not have any affinity towards this iron response element. It cannot bind to it. Okay, if it is not binding, it is not blocking the cleavage and the stability should reduce. Okay. Now the last statement says, when iron is scarce, then only this iron response element binding protein will bind to and stabilize the mRNA that encodes such and such. Okay, so here definitely when iron is in less amount, so iron will be utilized for some other things. It won't be available to come and bind to this protein. So now what we have the apoprotein. This apoprotein without the iron will have the ability to come and interact with this iron response element. Okay, as it is suppose binding here like this, what it will do, it will not allow the RNAs to gain access to this area and cleave this area. Okay, and that way they are maintaining the stability of the mRNA. Okay, so this one is also true. So which one is the correct option? Fourth one is the correct option. Okay, so you have to remember this particular mechanism. So iron response element binding protein is working like a cap. If you remove the cap, the area is exposed, it is degraded. If you have the cap, the area is hidden and it is not degraded. Okay, you have to remember something like that. Okay. Yes. Now clear, everyone? Okay, yes, good morning. Yeah. Yeah, what about Harshita and others who were not able to answer this? Is it clear? Okay. Okay, fine. Signaling, okay. True. Acha, which is true. Paracrine enabling communicate over short distances. Synaptic autocrine, okay. Endocrine signaling via hormones, okay. Yeah. Are you saying or option C, right? Okay, fine. Option D. No, read the option. You have to find out which is the correct statement. Okay, just see which one is correct statement over here. Yes, Arita? See, what is first one? Direct signaling is a key feature of only plant cells. What is the example? Direct signaling in plant cells, it can happen how? Yes? Plasmodesmata? Okay. So, in case of animals, we have what? The gap junctions. So, that means direct signaling is possible in both plants and animals. So, this statement is actually not true, completely not true. Okay. Because it is saying only plants. So, only one is not correct. Other thing is correct. Okay. So, this statement is again not the correct statement. Then, so this is wrong. Then we have paracrine signaling. Neighboring cells communicate over shorter distance. That is what happens in paracrine signaling. Signals will not be active when they diffuse after a particular distance. Okay, so this is correct. Next, synaptic and autocrine signaling are synonymous. Is it so? No, because autocrine is the cell which is producing the signal itself having the receptor. Okay, but in synaptic, it is one cell is releasing, another cell is receiving. Okay, so these are not synonymous. Okay, next, endocrine signaling via hormones. Okay, needs the signaling cells to express the receptor to bind the hormone. Signaling cells will express the receptor. No. That means the cells which are producing the signals, they will not have the receptor. The target cells will have the receptor. Okay. So if it is like that, then it will become autocrine signaling. It will never remain the endocrine signaling. So again, this statement is not true. Only statement which is correct is option C. Okay. Yes. Now clear, Meghna? Yeah. Okay. Yes. What about Neelam? Clear for you? Okay. Fine. So try to remember all the junctions and all. Okay. You might get questions based on these are very simple and direct questions. Okay. Next one. Yes. Which one? Option C. Okay. Yes, Arita? You chose B. But what do you think now? Okay, you think. I'll give you time to think. Option C. Yes. Anyone else online students? So what happens here basically, yes, you have a G protein coupled receptor. So this is the plasma membrane. You have a 7-pass G protein coupled receptor. Now here is a ligand binding domain and here is a G protein binding domain. Okay. Now, if you want to stop it from working, there will be a G protein coupled receptor kinases which will come and phosphorylate. Okay, and immediately this will be working as a docking site for the another protein, that is the arrestin protein. Okay, now this arrestin protein will block the binding of this G protein to this particular cytoplasmic domain. No more binding of the G protein, no more activation of G protein, no more signaling will take place. Okay, and additional to it, what it will do, it will lead to the internalization of this receptor by forming a endosome. Okay, receptor will move inside and no more it will be able to associate with the ligand. Okay, so here then based on this, which statement should be correct? C, binding to the phosphorylated receptor, preventing them from interacting with the G proteins. Okay, so only this statement is the correct statement. Other statements say that, see, binding to receptor, preventing them from phosphorylating themselves. G protein receptor, they don't phosphorylate themselves. They don't have that enzyme. Okay, that is the feature of the enzyme linked receptors, not this receptor. So this statement is not true. Phosphorylating receptors, preventing them from interacting with G protein. So arrestin is not phosphorylating, a kinase is phosphorylating. So this statement is again not true. And binding to phosphorylated G protein, this is again not true. It is saying arrestin binds to the G protein, but actually it binds to the receptor. So this is not true. Okay. Now I guess clear for everyone. Any doubts, online students? Okay, fine. See, the desensitization will be through all this. Okay, there will be internalization, there will be further cleavage. Okay, or there could be just sequestration for some duration. So all this becomes the part of the desensitization. Okay, Anjan? Yes, next question. Yes, CM error, right? Okay, yes. Option 4 is the answer over here. Okay. So that's why you have to remember some of the genes, okay, which are involved. Yes, inherited retinoblastoma requires dash number of mutations. Which one? So there is requirement of 2, yes. Then only it is going to express the condition. Okay, so yes, answer is option 2. Fine, good. Everyone have answered it. Okay, yes. Okay. Monocytes, Kupffer cells, mesangial cells are the different sites of the macrophages, right? Yes. So only endothelial cells are not the any type of a macrophage. Oh, Madhumanti, no, please recheck your concepts because mesangial cells are again a type of a macrophage. Okay, fine. So just check. I have already given the tissue location and the names. Just check that part, you will come to know. Okay, I guess it is somewhere, I don't remember exactly, okay, I should not comment on this. Okay, so mostly monocytes will be in the blood, Kupffer cells will be in the liver, mesangial in kidney, I am not sure of it. Okay, in kidney, right? Okay, I was confused between microglia and the mesangial. Microglia is in brain, right? Yeah. And endothelial cell is a different type of the cell. It's not exactly the part of the macrophages or the monocytes. Okay? Yes. Not a problem. Okay. Next we have, yes, which of the following is not produced? Yes, NO is produced by what? Arginine pathway. No, what is the enzyme actually? I want to know the enzyme. See, here we have to just remember which enzyme is giving rise to which radical. Okay. So here you said yes, NO synthase. Very good, Anjan. So it is the NO synthase which will give rise to the nitric oxide Other things, whatever are mentioned here, all these come through this the NADPH oxidase function. Okay, that's it. So this is all about the part B questions. Okay, so clear? All of you are clear about these questions? Online students? Okay, fine. Okay. There's a graph, I guess. Okay, just read it. I'll show the graph. Okay, let me read also. Read the question? Yes, everyone, online students. I'll show the graphs now. Okay. Okay, yes, option 1. Okay, because here we are silencing only one pathway, the other pathway is still working. Okay, so that is why a more appropriate answer is the option 4, the option 1. Sorry. Okay, 1 and 4. Yes, I know. Okay, so we read here. The question says that you have some cells. Okay, those cells we can trigger the apoptosis. How? By giving some X agent. Okay, some drug you are trying to develop, and that drug you are testing. Okay. So now we have what? We have two pathways of apoptosis, okay, intrinsic and extrinsic, and there are some initiator caspases, right? So for extrinsic it is 8 and for intrinsic it is 9. Somehow we are trying to only suppress the intrinsic pathway. Okay, so what we are doing? We are introducing the siRNA and we are trying to silence this caspase 9. So that siRNA will go on degrading all the RNAs which can produce the caspase 9. Okay, so that means only one pathway is functional. That is the extrinsic pathway, not the intrinsic pathway. Okay, so what will happen if we provide the same drug in such a situation? Okay. So this open box gives us the cell where no change has been done, no transfection of siRNA has been done, and the striped box is where the transfection has been done. Now, what will be the scenario if for both the cases we put the X agent? Okay, so ideally no change here, right? So it should give us both the extrinsic and intrinsic pathway of apoptosis. So there should be almost the highest amount of the percentage of apoptosis, maybe 100% or 99%, whatever is possible, that will be taking place. Okay. But in the next one, what do we find? There is one branch which is not working. Okay. So ideally, apoptosis should occur, but it may not be to the fullest extent. Okay. So maybe somewhere near 50% or 60% we can get the apoptosis. Okay. But what about this? This shows that apoptosis has lowered a lot. That means not at all, or maybe only very negligible amount of apoptosis is taking place. So maybe only 10% of apoptosis is taking place. That is not going to happen because we are not going to suppress the extrinsic pathway. We are only suppressing the intrinsic pathway. So at least 50% of the apoptosis should be occurring. Okay. But what about the other graphs? Here it shows that both are having same. That cannot happen because we are only transfecting one of these cells, not both of these cells. Okay. So here there could not be suddenly a decrease. Okay, that is not possible. Okay. Even if there is a decrease, okay, or maybe even if it is because of the X is not able to bring all the cells to move into apoptosis, then also in this case it should be lowered. because here we are suppressing, so it cannot be completely 50% like the other one. So this is again not correct. Here it cannot be more than the one which is not treated, because we are disabling here, not here. Okay, so that is why option 1 is the correct one over here. Okay, yes, fine. So I guess it is clear for all now. Yeah. Yeah, yes. Yes. Correct. Option 2. Neelam, do you mean option 2? Yes, what about others? Online students? Only we. Okay, only we means third one. Okay. Option 2, option 3, option 2, ABC. Okay, fine. See, what is happening is there is a person who underwent some treatment of surgery, chemotherapy for the breast cancer. Okay, and it was found to be subsiding for the initial treatment, but after 5 years the tumor was found to come back. Okay, and it was found to be more aggressive in nature. Okay, so like it was no longer responding to the treatment like that. Okay, and maybe it was more invasive and going to spread. That type of nature was seen in the new tumor. Okay, so what could be the possibilities that even after treatment, how come this type of relapse can occur? So first statement says that some chemo-resistant cells were persisting within the tumor even after the therapy. So, see, you surgically remove it. Still, some of the cells will be removed, because you cannot singly isolate all the cells, right? That is not possible. Surgically is not like microscopic view. It is like more macroscopic view, right? So that means some cells might have remained behind. Those you try to treat by giving the chemotherapy. Now what we have learned is cells will gradually become resistant. Okay, so those cells which were resistant, they remained behind. Okay, and many cells they died. So maybe these cells were for a long time they were suppressed. Okay, after nearly 5 years, see, when you have something like this, you altogether don't stop the treatment. You continue to have the doses, but in maybe lower amount. Okay, so maybe when it was found to be getting some low dose and it was getting the condition to survive, then it again started to grow. So that might have happened. Okay. Then option B says a population of poison cell existed. That means those cells which were not dividing. Okay, they were simply sitting in the G0 phase. Okay, and that is why they were able to restrict whatever was the unfavorable condition at that time. Okay, they were somehow behaving like a spore. Okay, and when the maybe the dosage of the chemotherapy reduced, the favorable condition came back, and then they were able to regrow again. Okay, that could be the another case. Then high ABC transporter expressing cells persisted in the breast during the chemotherapy. That could have happened. Okay, so these three possibilities look okay. Okay, to explain the situation. Now, what about the last one? The breast tumor cells, which may have migrated to other tissues, return to the breast immediately after the chemotherapy was terminated. So, it cannot happen. Okay, they might be locating in some new region and they can start a tumor in the new region, but again coming back to the same location. Okay, it's something looks like very melodramatic. Fine, so this is not correct. These are more probable type of the situation. Okay, or the reasons why these type of relapse was observed. Okay, yes. So clear about all this question, everyone? Okay, fine. So I guess the question paper was easy, right? This time, yeah. So your marks also would have improved. It should improve actually. You should keep a constant watch on it. Okay. Fine. So let us now move into the antibody structure. Yes. What is the basic structure of antibody? Yeah. Okay, two heavy, two light chain. Okay. There is a constant region, there is a variable region, there is a hinge region, right? Okay. And? Yes, very good, very good. So there is a disulfide linkage between the heavy chain, light chain, and that is why it gives rise to a dimer formation. Okay, and altogether the structure will resemble the Y, right? So that is what is the basic structure of the antibody. Okay, what the variable region does? Okay, yes, variable region has CDR, okay, good, and it is binding to the epitope of the antigen. Okay, so we can say antigen binding sites. Okay, and what is the role of the C? Constant region. Yes, it gives a biological function to the antibody. Okay, it can go and in turn bind to some specific receptors. Okay, yes. So we will see that part in detail today. Okay, we have seen the broad structural view of the antibody. Now let us look at the fine structure. Okay. Okay, yeah, this experiment is also very important. Please don't forget the molecular weights here. Okay. So what should be the molecular weight of these two? Yeah, and this one? This one? Okay, and these two? Yes. And what is the enzyme? What is giving rise to this? Pepsin, papain, and it is the beta-mercaptan. Please remember this, okay? So these are some of the important things that you should remember. Okay, fine. Okay, now looking at the fine structure. So fine structure, yes, gives us the idea of the domain folding of the immunoglobulin. So it's showing some domain-like structures. So some regions will be folded into the domain, and there will be some linker region between the domains. Okay, so these domains are actually having the beta strands. Okay, so there will be actually beta strands or two beta sheets, okay, which will be having multiple beta strands. Okay, so almost we have some nearly 8 to 7 beta strands which will be arranged anti-parallelly to each other. Here also nearly we have some 8 or maybe 5 to 6 beta strands which will be arranged anti-parallelly. Okay, so once they are arranged like this, in between there will be some linker region. Okay, so these are the loop-like region. Okay, this loop-like region are allowing the linking of. Okay, let us focus here rather than that one. That is a 3D structure. The below gives us a 2D structure. Okay, so it is like the N terminal or the N terminal linking to the C terminal. So like this, the linking will be taking place with the help of the loop. Okay, and they will be arranged, the two beta sheets, they will be arranged in such a manner that the two beta sheets will be linked by a longer loop. Okay, so long loop will be there which will be linking them, and in between there will be a disulfide linkage. Okay, the disulfide linkage will be present between two amino acids which are approximately 60 amino acid distance apart. Okay, so that way the entire immunoglobulin fold will look like. Okay, so this particular folding pattern is called as the immunoglobulin fold. So we have already studied the immunoglobulin superfamily of CAM molecules. Actually, they have this particular folding pattern. That's why we call them as the immunoglobulin superfamily. So not only CAM molecules like this, so many other molecules are there which follow this same pattern of folding. Two beta sheets held together by a disulfide linkage, and the approximate distance is 60 amino acid between the disulfide linkage. Okay, but the total domain will be of approximately 110 amino acids. Okay, that will be the total domain structure or the composition or length we can say. Okay. Now the first domain is the variable. Okay, variable region. So for heavy it is VH, for light it is VL. Okay. Now if we compare the other domains, other is a constant region, right? So when we compare this particular folding pattern in the variable region and in the here in the constant region. So this is variable, sorry, this is the constant region. Okay. Now here what do we find? The variable region is actually more extended. Okay, it is more longer in size. Okay, how? If you count the number of the beta sheets over here, it's only 3. Okay, but here it is actually 5. Okay, so that means we have 2 extra beta strands present over here in this particular beta sheet. Okay, whereas it is much more smaller. So this is what gives it a extended look. Okay, or it is making it more extended in structure. Now it has to be extended. Why? Because it has to hold the antigen. So it cannot be smaller structure. Okay, so if we see, we know that there are some CDR regions, right, which are hyper variable, right? So these CDR regions are actually located in the loops. So this is the location, this is the location, and this is the location of the CDR. Okay. Now CDR are hyper variable. They are the one which will be holding the antigen. So that is why they need to be flexible, so that they can mold themselves, they can be mobile, and they can fold or they can move in such a manner that they can hold the antigen. Okay, so that is why it will be the loop region which will be having the CDR. Okay, now what about the rest of the region? Rest of the region job is to give a framework. Okay, to give a support to the CDRs. Okay, that is why we call this as a framework region. Okay, so most of the cases when you try to look at the, or you try to superimpose the 3D structure of this framework region, you find it was more or less superimposing. Okay, there is not much orientation differences, but the major orientation differences will be lying in the CDRs. Okay, so CDRs will have the hypervariable regions. Okay, the amino acid sequences will be rarely matching in this region. Okay, along with that, the orientation of the loops will also be different because they have to bind to different epitopes, right? So that is the nature of this framework region and the CDR region. So always remember, okay, last class itself I told, one light chain variable region, one heavy chain variable region together will form the epitope or the antigen binding site. Okay, it is not that only a light chain will have a separate antigen binding site or a heavy chain will have a separate antigen binding site. It's not like that. Okay, so 3 CDRs will come from this light chain and 3 CDRs will come from the heavy chain, and then all the 6 together will hold the antigen. Okay, so let us look. Okay, in this diagram we can understand. So if we take the analogy of the human hand, okay, so CDRs are these 3 fingers. Okay, so these 3 fingers which are actually coming in contact with this apple. So this apple is the antigen over here, okay, or the epitope, fine, and this part of the hand is the framework region. So it is giving the support to the CDR, okay, so that it can change its orientation. Finger can move in any orientation and it can accommodate the antigen, but this support region will be more or less rigid in its form. Okay, it is just giving it a framework to stand or to move. Okay, so here this is the actual view. Suppose we have an antigenic epitope. Now we can see epitope is having a specific pattern of folding, right? Now to match this, the CDRs will also fold to have a complementary binding site. Okay, so it is the variable region CDR2. This is the variable region CDR1. This is the variable region of light chain CDR3. Okay, so this variable region belong to the light chain. And this side we have the variable region of the heavy chain. So this is CDR3, this is CDR1, this is CDR2. 2. So like this, the CDRs will rotate in any orientation, okay, and they will start to give rise to the structure where the antigen, the epitope can fit inside, okay. Actually, the interactions, if we talk about, the interactions will be the non-covalent interactions, okay. That means the hydrogen bonding, the hydrophobic interaction, the salt bridge, okay. These type of interaction will be there, but they are not, and mostly, only some of the amino acids are going to interact, not the all amino acids. In CDR also, maybe 2-3 amino acids. In one CDR, 1-2-3 amino acid, another CDR, again 2 or 1 amino acid. Like this, the interactions will happen, okay. So as a result, what can happen is the interaction may not be very strong, okay. So what you need? Complementarity. When you have structural complementarity, it increases the binding affinity, okay. That is why you need mobility, and that is why CDRs are present in the loop region, okay. At the same time, when we studied about the epitope properties, we studied that the epitope has to be accessible, epitope has to be hydrophilic, and it should be a mobile peptide. Again, because epitope also has to fit inside into the binding site, okay. This complementarity in structure is what is making the binding more stronger in nature, okay, along with the non-covalent interactions, okay. So if we compare the antibody and the T cell receptor, antibody has a higher affinity compared to the T cell receptors, okay. Antibody alone can bind the epitope very tightly. T cell receptors cannot do that. They need lot of help, okay. So that is the major difference between the antibodies and the T cell receptors, okay. Whereas here we see the constant region is smaller, not that much extended. You have nearly 4 beta strands over here and 3 beta strands over here, again arranged anti-parallelly, and there are the loop-like regions which is joining them, okay. So this is but here it will have some other jobs to do. Variable region have loops will perform the job of the binding, okay. Framework will give us support, but constant region domain will have some specific roles, other specific roles to serve, okay. This part is clear, how antigen binding site is created with a heavy chain and a light chain CDR. So 6 CDRs together make up the antigen binding site, okay. Okay, yes. So that means in an antibody, how many antigen binding sites are there? How many? Yeah, tell. Yes, because we have one here, light chain, heavy chain, another light chain, heavy chain pair is present over here. So that means 2 binding sites are there. Okay, then altogether how many CDRs? Yes. So these concepts should be clear to you, okay. So each heavy chain or light chain, each will have 3 CDRs. Together they form 1 binding site, okay. So altogether 2 binding sites, and altogether 12 CDRs, okay. Yes. So that is the job of the variable region of both the heavy chain and the light chain. So next we have another immunoglobulin fold, okay. This creates the second domain, okay. Now this domain, approximately 110 amino acid. In case of light chain, there is only 1 constant domain, okay. And in case of the heavy chain, there could be 3 more or 4 more constant domains, okay. So the one which is immediately present next to the variable region, now what is its role? Fine. So first of all, let us talk about first of all, let me clear the slide. It looks so bad. Yes. Now what is the role of the CL? CL will be again either of kappa or lambda, okay. So it actually increases the variability, okay, variability of the antibodies, okay. Antibody can be of kappa type, antibody could be of the lambda type. Okay, that is one job, okay. So we need the variable types of antibodies to target the numerous pathogens which are available throughout the environment, okay. So that's why variability is the key for antibodies. So CL gives the variability, Along with that, what do we find? It is needed for this one as well as this one, CH1. Okay, it is needed for extending this variable region. Okay, so that it can easily reach out to those epitopes which are somewhere buried deep inside or something. Okay, so that means it is allowing it to avoid any steric hindrance. Suppose this region was not there, that means it will be immediately present next to the hinge region. So then suppose you have two epitopes present side by side, then what will happen? Like this, the binding site will be very small. This is the hinge region over here. Okay, so what will happen? This site maybe go and bind here. Okay, but this site will not gain the access to this region. Fine. So that means it will not be able to bind because of steric hindrance. To avoid this, you are little bit extending the binding sites by providing this scaffold-like structure over here. Okay, so this is needed for extending the binding site so that it can reach out to the different epitopes at the same time. Okay. Now, what is the job of the next region? Now, after this CH1, okay, you can have hinge region. So, based on what type of the heavy chain you will have a hinge region or you may not have a hinge region. Okay, so mostly if it is gamma, if it is the alpha, and if it is the delta, in these three cases you have a hinge region. Okay. Now, what is the job of hinge region? Okay, hinge region speciality is that it is rich in proline, it is rich in cysteine. Okay, that is why you have numerous disulfide linkages over here. Okay, so one more thing which I forgot to tell about this C domain is the linking between the heavy chain and light chain will be because of the disulfide linkage between the CH1 and the CN. Okay, this part I forgot to tell. Okay. So coming back to the hinge region. Hinge region is the part. Yes, fine. Hinge region is the part where you have more of proline and you have more of the cysteine residues. So cysteine is needed for disulfide linkage, which will help in forming the dimer. Okay, dimer structure. Okay. What else is the job of the hinge region? Okay, let us imagine we have one epitope over here. Okay. There is a pathogen and there is another epitope somewhere over here. Okay. Now, if this was the constant structure of the antibody, this side maybe go and bind, but this side cannot go and bind, right? So, the importance of hinge region is, now proline gives, whenever proline is present, it gives you an extended structure. Okay, a linear extended like structure like this. Okay, and then you have some globular fold. Again there is a linker, then you have a globular fold. Same happens here also, you have a extended structure like this, globular fold, and again one more globular fold. Okay, now this will allow the movement in this manner. Okay, so it gives you stretchability or flexibility. Now as a result, now if this side goes and binds here, this side will be bending and then it will reach out to this epitope. Okay, so hinge gives the flexibility. Not only it is allowing the binding between the two dimer, giving rise to the complete antibody structure, it is also giving flexibility. Okay, but again the flexibility of hinge region can be differing. Okay, that part we will understand when we will go into the details of the sub isotypes. Okay. So for now, remember, when you have hinge region, it gives you a better flexibility. Okay, but what about the other two types, the mu and the epsilon? Okay, when you have these type of heavy chains, then there is no hinge region, but in that place you have a CH2 domain. Okay, CH2 domain will again have the same type of the immunoglobulin fold. Okay. But what is the role exactly, that is not very clear. One role is that it is linking the CH1 to the CH3. Okay, that should be the role of CH2. But apart from that, what could be the other biological role, that is not very clear. Okay, because it is not giving any flexibility like the hinge region. Then how it could be, having any other role, that is yet to be discovered, or maybe yet to be there under study probably. Okay, so that should be the additional feature of those heavy chains where there is no hinge region. Okay, next we have the presence of CH2 domain in case of the gamma, delta and alpha, but in case of mu and epsilon it should be CH3 domain. Now what is the importance of this domain? This is the domain which gives it the biological activity. Okay. Now one of the major feature here is, okay, it is having the polysaccharides attached to it. Okay, so this is the one which will be glycosylated, and as a result it will make this domain more accessible to the aqueous environment because it is having some sugar molecules attached to it. Okay, as it is more accessible, that means complement protein can come and bind easily to this side. Okay, even different types of the FC receptors are there, they can come and bind. Okay, so FC receptors present on the NK cells, present on the neutrophils, present on the macrophages, RBCs or whatever it is, those receptors will be again able to access this particular region of the antibody. Okay, so that is the major role of this CH2 or the CH3 domain. Lastly, we have the CH3 or the CH4 domain. What is its function? Its function is to decide whether this antibody will be expressed in a membrane associated form or it will be secreted out. Okay, how it is deciding it? If it is secreted form, this region will have hydrophilic amino acids. Okay, so there will be one alternative splicing mechanism. Because of that, some exons will be removed, some exons will be added. Okay, so what do we find? If it is secretory form, then the exons which are coding for the hydrophilic amino acid, they will be added to the C terminus. Okay, so as a result, they will be released out. They will never get incorporated into the plasma membrane. Okay, but if it is a membrane bound type of the antibody, then there will be a transmembrane domain. Okay, which will be hydrophobic in nature. As a result, it will be, when it is passing through the plasma membrane, transmembrane domain will allow it to sit at the plasma membrane, not get secreted out. Okay, so this is the importance of the last domain, the CH3 or the CH4 domain. Okay, now clear? What are the functions of each domain? What is the purpose of this black band? Okay, Madhumanti, this is a disulfide linkage. Okay, it's a disulfide linkage and it is what is giving it the globular structure. Okay, fine. Yes, so always remember variable region CDR participating in binding the epitope, the framework region giving a support, the constant region 1 is giving it a extended form, okay, allowing it to reach to the different epitopes. Okay, hinge region gives its flexibility. Then CH2 or CH3 domain is needed for biological function. How? It will be having the oligosaccharides, and this oligosaccharides will be making it more accessible to the complement, making it more accessible to other type of receptors. And what about the CH4 and CH3? They will either have a hydrophilic C terminus. If it is hydrophilic, it will be secreted out, then it moves into the bloodstream, into the tissue fluid. Okay, and if it is having a hydrophobic transmembrane region, then it becomes membrane bound receptor. Okay, so that will be deciding what way the expression of this antibody should take place. Okay, yes, so these are the major roles. Try to remember them. Okay, so clear everyone, right? Online students? Okay, fine. Now, what are the different isotypes? So, isotypes we classify based on the heavy chain, right? So, IgG mostly having what? Gamma type of the heavy chain. But it can have different subclasses. Gamma 1, Gamma 2, Gamma 3, Gamma 4. And what it can do? It can combine with either kappa light chain or it can combine with the lambda light chain. So, based on whatever it is combining, the entire molecular formula will be 2 heavy chains of gamma type, 2 light chains of kappa type, or 2 light chains of lambda type. Okay. In the same manner for IgM, it will be the mu. There is no subclass here, and here we can have 2 heavy chains of mu, 2 heavy chains of kappa, or 2 heavy chains of mu, or 2 heavy chains of lambda. But this could be, this entire tetrameric structure could be repeated. Okay, that means it can form some polymeric structures. Okay. So that is what is represented by N. So either one antibody of IgM could be present, or it could be 2 dimer, trimer, tetramer, pentamer, even hexamer, heptamer is also possible. Okay. So that those are also found to be present. Okay. Then what else? IgA. IgA will have the alpha heavy chain. The subtypes are the alpha 1 and alpha 2, and again the combination will be either with kappa or the lambda. So here also we can have polymerization. So either dimer, trimer, or tetramer possible for the alpha IgA. Okay. Now what about IgE? IgE is carrying the epsilon, kappa or lambda again. Okay. And IgD delta, there is no subclass, kappa or lambda again. Okay, so molecular formula will be respectively same as that of the other types. Okay, so this is how the composition of the different types of the antibodies will be having the composition. Fine. Next we have functions of the antibody. Already we have discussed. Okay, opsonization, that is to coat the pathogens and then help in the phagocytosis. Neutralization is just to bind to the pathogen and clear it out from the system. Okay. Best example is the mucus secretion. Okay. In mucus secretion, you can have IgA, right? So what the IgA will do, for example, this is the cell surface, and this is the pathogen. Pathogen wants to attach to the cell surface. Okay. But the mucus secretion already has the antibodies. What it will do, antibody will go and bind to the sites with which the pathogen can attach. Now pathogen sites are blocked, attachment sites are blocked, it cannot attach. As the mucus secretion will flow, it will be taking away the pathogen. Okay, so that is what is neutralization. Same thing can happen inside the body also, where in the blood the pathogen could be neutralized and then the pathogen could be taken to liver. From there it could be passed on to the phagocytic cells. Okay. What else? We can have antibody activated complement. That means antibody once goes and binds to the pathogen, it can, with this CH2 or the CH3, mostly it should be CH2, it will be participating in the complement activation. When complement is activated, it can directly lyse this particular bacteria or it can help in the phagocytosis. Okay. Next we have antibody directed cell mediated cytotoxicity. Here are the NK cells. Okay. So IgG receptors are there, right? CD16. So NK cells will recognize the recognize the IgG with the CD16, and then it will start releasing the enzymes and other things which will kill the pathogen infected cells. Okay. So these are the major functions of the antibody. Okay. Otherwise, we can also say that they will be constantly patrolling the blood circulation, the tissue fluid, and looking for the pathogens just to tag them. Okay. And rest of the job will be done by other things. Okay. Yes, so clear about the function, everyone? Online students? Okay, fine. So you might get some match the following type questions based on the different types of the antibody. So we will go into little bit details of the different isotypes of the antibodies. Okay, these are the structure. This is IgG, IgD, IgE, IgA dimer. This is the pentamer of IgM. Okay, let us see one by one. This is a table which gives us an idea of the basic differences. Okay, we will focus on this. Okay, so immunoglobulin G. Okay, it is the most abundant class of the antibody which will be present in the serum. Okay. And it is supposed to be, making about 80% of the total serum immunoglobulin. And we have two types of responses, the primary response and the secondary response. So usually IgG is produced in the secondary response. So this is the most abundant type of the antibody produced in the secondary response. It is having 2 gamma heavy chain and it is having either 2 kappa or 2 lambda light chains. Okay. Again, the gamma can have different type of sequences. Okay, so based on that, we classify as G1, G2, G3 and G4. Okay, there will be mainly differences in the hinge region. Okay, otherwise some other regions can also differ, the constant region, but the hinge region will be mainly differing here based on the length of the hinge region and the flexibility as well as the disulfide bonds. Okay, let us see. Okay, so these are the 4 different types of the IgG. What is differing here? Mainly you can see difference in the length as well as the difference in the disulfide bond. So here the total length of the hinge region is 15 amino acid. Okay, and it has got only 2 disulfide linkages. So it is highly flexible. Okay. It is showing the highest flexibility. Okay. Then we have IgG2. Okay. Here the hinge region is smaller, actually only 12 amino acid. Fine. Same for IgG4 also. Here also length is 12 amino acid. Now the number of disulfide linkages 4. Here also 2 to 4 linkages are possible. Okay. But still the flexibility of this is found to be lower than this. Okay. So that means here we can say the flexibility of IgG2 is somewhere intermediate between these two. Okay. Or somewhere lower than these two. Okay. So it is intermediate to this one. Okay, I incorrectly said it is intermediate to Ig3. Okay, that means Ig3 is not flexible at all. Lowest flexibility is shown by Ig3, and in between you have this 2 and 4. But 4 is still more flexible compared to the Ig2. Okay. Next we have the IgG3. Here the longest hinge region, approximately 62 amino acid, and the number of disulfide linkages are approximately 11. So more disulfide linkage, that means more rigidity. Okay, so that is why here the lowest flexibility will be seen in case of the Ig3. Highest flexibility in IgG1 and here lowest flexibility. Next to IgG1 we can have IgG4 and then IgG2. Okay, so this way the flexibility will be differing, and based on this we subclassify. Okay. Apart from this, there will be also some minor differences in the heavy chain, okay, sequences, but those are not that much prominent. Okay. Now, if you want in detail, you can refer this particular paper. Okay, there more detailed view of these subclasses are given. Okay.
IgG4 is more flexible than IgG2.
Yes. Yes. Okay. Actually, you will find in the hinge region there will be more difference in proline concentration. So based on that, it will restrict the movement actually. Okay. So based on that, you have some differences in the flexibility. Fine. Now, what are the roles of this IgG? Okay. So already we understood this is the major type of the immunoglobulin which is produced in the secondary response. Okay. Apart from that, IgG1, IgG3 and 4, they have the ability to cross the placenta. That means they are the major antibodies which are giving protection to the infant. Okay. Next, IgG3 is the most effective complement activator. Okay. So always remember, all the antibodies, they do not activate the complement. Okay. So here it is the IgG3 which is activating the complement, followed by G1 and G2. Okay. Now we have G1 and G3 having the ability to bind to the Fc receptors on the phagocytic cells and then mediate the opsonization. Okay. Now G4 can have some but very low affinity towards the Fc receptors. Okay, so these differences are mainly because of the flexibility order is no, that is in this is the increasing order, we can say. Okay, 3 is the least flexible, then comes the 4, then it should be 2, then it should be 4, and then should be 1. Okay, fine. Neelam, what you are saying is actually the reverse of it. Okay, so it is just a sec. See here, this is more flexible, then comes the 4, then comes the 2, and then comes the third one. Okay, it is the least flexible, this is the highest flexible, and these two will be in between. Even though 4 and 2 are almost same in structure and all, but still 2 will be less flexible compared to that of the 4. Okay, now clear? Okay, fine. Okay, so that these are the major jobs of the IgG. Based on again the flexibility and all, the jobs can little bit differ. Some IgG will be giving more preference to one type of role, and another will be giving more preference to other type of role. Okay. Next we have immunoglobulin M. Okay. So IgM, this is making 5% to 10% of the total serum immunoglobulin, but it is the major antibody which is produced during the primary response. Okay, and even the first type of antibody produced inside a body of or maybe inside the neonate. Okay, so this is the first type of the antibody which will be generated. So when we talk of the B cell receptors are actually IgM. Okay. So when they are secreted, they could be of other forms. Okay, not always IgM. They could be IgG, they could be IgA, they could be IgE. Okay, but mostly when they are working like receptor in the naive B cells which are not activated, then it is the IgM. Okay, and it is also having the ability to get secreted. Okay, when it is secreted, it is preferring the pentameric form. Okay, so pentameric form, the structure already we saw here. Okay, so this is one of the IgM. There is no hinge region. See here, there is an additional constant region too. Okay, now like this, there will be 5 IgMs which will be arranging, and the linkage will be again a disulfide linkage in this CH3 domain and CH4 domain, and there will be additional J polypeptide. Okay, this is called as J chain. This will further link the two of the antibodies. Okay, now this is very important. This actually acts like a stabilizing feature of this pentameric structure. Okay, next if we talk about the functions, the first immunoglobulin to be produced. Now because they are pentamer, they can bind to more number of epitopes, right? Because there will be in pentamer, how many binding sites will be? 10. Yes. So they can bind to 10 epitopes. Usually when the molecules are smaller, then only 10 epitopes. If it is a huge molecule, the steric hindrance will be there. So 5. Okay, maximum 5 or maybe 6. That many epitopes it can bind. Okay. So that is why it is found to be very efficient, even more than the G, to activate the complement. Because in complement activation, you need such type of antibodies which can have multiple binding. Okay, as well as they can bind to those type of epitopes more efficiently which are repeating in structure, like the peptidoglycan cell wall. There is a NAM, repeat. Okay, then you have flagellin. You have flagella where flagellin protein will be repeating. Okay, so those structures which are made up of repeated monomeric units, those type of antigens will be better bound by the immunoglobulin M. Okay, so these are the major features of the immunoglobulin M. Next we have immunoglobulin A. It also makes up nearly 10 to 15% of the total immunoglobulin in this serum, but its more important role is in the external secretion. So tears, milk, okay, saliva, then urine, okay, gastrointestinal fluid secretions. All these secretions are going to have the IgA. Okay, its job is to protect the mucous membranes. Okay. Now, and in case of baby, it is just transferring the through milk, transferring the IgA directly to the baby. Okay. Now, what are the different polymeric forms? Again, we have dimer, trimer, tetramers are possible. They are all stabilized by a J chain. There is an additional feature called as secretory component, because of which we call it as a secretory IgA. Okay, this secretory component is again a polypeptide and approximately 70,000 Dalton in molecular weight. Okay, let us see the structure. See, this is one IgA, this is a second IgA. Now here we have a interlinking J chain, and cross-linking these two is the secretory component. Okay. Now actually secretory component also gives stability. Mostly you will find it is almost covering the hinge region, the J chain, and again another hinge region. So these are the regions which are more susceptible to protease degradation. Hinge region is again more susceptible to protease degradation. So what it is doing, it is actually giving protection, it is blocking against the protease degradation so that it can remain in the secretion in active form for a long time. Okay. Now what the pathogens do? Pathogen will actually try to evade the IgA by secreting proteases so that it could be cleaved. Okay. So that is the way the pathogens try to evade the IgA molecules. Okay. Now how this secretory component is attaching to this IgA. Okay. So usually let us consider this is the gastrointestinal lumen. Okay. So we have the epithelial cell, this is the apical surface, this is the basal surface. Okay. Now plasma cells will be somewhere here, they will be secreting the dimeric form of the IgA. So initially the secretory component is not present in the dimeric form. Okay. Now what it will do, because it has to be released into the lumen, then only it will give protection to this area, right? So it will go and bind to some receptor which is expressed on the basolateral surface. Okay, this is called as the poly-Ig receptor. Why? Because it is binding to the polymeric form of the immunoglobulin. Okay, so this poly-Ig receptor has an extracellular domain which has got this 5 repeats. Okay, again these repeats are having the immunoglobulin like folds. Okay. Now these repeats will be interacting with the here it is IgA, right? So it is CH2 domain. Okay. So CH2 domain of one of the out of the dimer, one of the antibody. Okay. And after this binding, immediately this receptor will lead to the endosome formation. Okay. Now this what will happen? This endosome will be moving across the cell and then it will be opening up on the apical side and it will be releasing the dimer. Okay. Now when this release is taking place, there is a enzymatic cleavage happening. So this part is cleaved off. So now the receptor itself is associating with the dimer and this is what forms the secretory component of the IgA. Okay, so the receptor helps in the transport. When it is actually getting released, it is facing the enzymatic degradation and that time it completely associates with the dimer and now it is a part of the secretory IgA. So this is how the secretion will be occurring in the IgA molecule. J chain, no, not exactly. J chain's job is to work like a linker, okay, and it gives stability. Fine, it gives stability to the dimer. Okay, fine. So clear this part, the function of IgA and how it is getting secreted. So try to remember this, small features. Okay, these, if you know this, whatever question comes, you will be able to answer. Okay. Then comes the immunoglobulin E, and this immunoglobulin E is found to be in very low concentration. Actually, if it is high, that means the person is suffering from some type of the allergic responses. So usually concentration will be 0.3 μg per ml, that much. Okay, and yes, this is the one which is going to mediate the hypersensitivity, the allergic responses. Okay. So how it is performing in the allergic responses, that part let us try to understand. Mostly IgE has to go and bind to the basophils, because basophils are the one which will release the components for hypersensitivity or the allergic response, and it is the mast cells again. So in the blood it is the basophil, in the tissues it is the mast cells. So immunoglobulin E should go and bind to these two type of cells. How it will bind? With the help of again the Fc receptors. Okay. So again the binding will be either at the CH2 or the CH3 domain. Okay. Once this binding takes place, it will allow the cross-linking and a signaling pathway will be triggered. Calcium increase, release of the vesicles. That is how the degranulation takes place. Let us see the diagram and understand. So here we have so somewhere maybe there is a plasma cell has secreted the IgE, okay. IgE has traveled through the blood, it has moved to the tissue fluid and then it has reached the mast cell in the tissue, okay. Now mast cell is having the Fc receptor on its surface. Now IgE will go and bind to this Fc receptor, mostly it is E, right, so it should be CH3, right, so CH3 domain. Through CH3 domain, it will interact with the Fc receptor. Now, usually what happens when two such receptors are cross-linked? How? Because of binding of an allergen or maybe any type of a pathogen. Okay. So once this one receptor over here, another receptor over here is cross-linked like this, there will be some phosphorylation or some activation of different enzymes that will start. Okay. So once the enzymes are getting activated, they in turn will lead to the secondary messenger formation. Ultimately, calcium concentration will increase. Calcium concentration increases, that will lead the vesicles to move towards the plasma membrane. Same as it happens in neurotransmitters. Okay, the calcium channel opens up, calcium concentration increases, the vesicles containing the neurotransmitter moves towards the plasma membrane. Same manner here also, vesicles which are carrying the mediators of the allergic response, they move towards the plasma membrane and then they fuse with the plasma membrane and release their contents. Histamine, heparin, okay, the platelet activating factors, so different type of factors will be released during this degranulation step. So degranulation needs the IgE. Without IgE, neither it can bind to the allergen nor the cross-linking can happen, and as no cross-linking, no signaling pathway will be getting activated. As a result, no release of the components. Now these components will start the inflammatory response. Histamine, vasodilation, okay, then there will be permeability increasing, okay, then platelet activating factor, again it is recruitment, it is recruiting the neutrophils and all, okay. So like this the inflammatory responses will start. These inflammatory responses are the one which is giving rise to the allergic reactions, okay. So this is how IgE plays an important role. Fine. Now but remember, normally IgE will also participate whenever there is a parasitic infection. That time also it will try to bind to the parasite and then recruit the cells to kill the parasite. Okay. But a normal condition we have this. Okay. Fine. Next we have the IgD, immunoglobulin D. Again, it is found to be in lower concentration only, about 30 gram per ml. Okay, only 2% of the total immunoglobulin. Now here, its function is still not clear. But what is observed is when you have the mature naive B cell formation, okay, the B cells which are yet to get activated. So I already told they will be expressing the IgM on their surface, right? But they also express the IgD. Okay. So maybe at this stage they can participate in antigen binding to activate the B cell. But after this they are not expressed. Okay. So only where they are expressed on the surface of a mature but naive B cells which are not activated. Okay. So maybe they are working like a co-receptor here along with the IgM. But apart from this, what could be the other function, that is not clear. So these are the five major types of the immunoglobulins. Okay, try to remember their basic function. You get match the following type questions. Okay, so on what basis we classify the G, M, and the other types? Okay, yes. So clear about these? Yes, online students. Okay, fine. Okay, yeah. So let us stop the discussion over here only. Okay, and tomorrow we'll continue with the rest. Okay, actually there are so many things in receptor. Okay, so you have to be patient because receptor structures are very, so many molecules are there, so many receptors are there, and structural details are very complex. Right? Okay. Okay, yes. So that's it for today's class.